Hi everyone, my name is Shaezmina.
And I'm Erin.
And we're seniors at the Lawrenceville School, and today we're going to be exploring how we can use machine learning to better understand complex biological processes.
So first we're going to give a little background about our research. So our study used the Briggs-Rauscher reaction. Basically that's a oscillating chemical reaction that is the result of multiple redox reactions. So it's exchanging electrons within it, which causes color changes between a blue, yellow, and clear phase, which we'll see in the video in a second. And it also causes varying voltages in the liquid, which makes it easy to track the oscillations with a voltmeter. And that's also connected, the Briggs-Rauscher reaction is also connected to biochemistry because the oscillations that we see in this reaction mimic some of the oscillating reactions that we see in the body, especially in the heart.
So here's one video of us running the reaction with a blank trial. So what you're going to be seeing in here is oscillations between the blue phase and the yellow phase, which is also oscillating in voltage. And I guess what's important to know, even if the video doesn't load, is that the reason that it's going to be changing between the two colors is because there are changing concentrations between the iodine and the iodide, and that's precipitating the oscillations going on. So we also performed an antioxidant study when we were looking at the Briggs-Rauscher reaction. So in between the phases and when it's changing colors, there are free radicals, which are basically oxidants, which are unstable molecules that have an unpaired electron that are floating around. And an excess of free radicals in our body actually causes a host of many diseases. But when you're able to put an antioxidant and put it into the Briggs-Rauscher reaction, what we see is it donates an electron to the free radical, and when it does this, it basically elevates and extends the period length between the oscillations in the phases, because it's extending the time in which the equilibrium is being reached and the shifting in colors is happening.
So next we can kind of talk about our research and our procedures. So first, our question was eventually what tea has the greatest antioxidant potential as evidenced by the Briggs-Rauscher reaction, and we theorized that cranberry would have it based on just the common conception that cranberry has high antioxidant potential. So there was kind of two parts to our study. We took an interdisciplinary approach. So the Briggs-Rauscher reaction is very sensitive with its initial conditions. So in order to best do this study, we wanted to find out what the best initial conditions would be. So we used a machine learning program that we wrote actually to find the best initial conditions. So what we did is ran 2 trials at 3 separate temperatures and 3 separate hydrogen peroxide concentrations, and plugged those data points into a machine learning program that we wrote, which then used a quadratic regression line and found the maximum of that and just suggested that as the new points that we could use. And so after we did a few trials and used the new suggested values, and the suggested value stabilized around a specific number, we were able to continue to explore.
And after we got the optimal conditions for the reaction to occur, so changing slides?
Yeah.
For the optimal conditions for the reaction to occur, we then use those for the antioxidant studies. So we studied 3 different tea types, cinnamon, cranberry, and green tea. And what we did is basically find 1 milliliter of those, dilute those in 100 mils of deionized water, and then we would add the tea suspension at the start of the second blue phase in the Briggs-Rauscher reaction. And basically what we would do is not only analyze with our own eyes, but also see the graph and see how the period was changing at the start of the second blue phase to the next phase. And we'll discuss that more in our results, which are really exciting as well.
So this was just our materials list. As we mentioned before, we used a voltmeter in the solution to track the oscillations. So next we can talk about our results. So as I just mentioned, we used a voltmeter to track the oscillations. So you can see how the Briggs-Rauscher reaction very clearly oscillates between the different phases. So here the y-axis is the voltage and the x-axis is the time. And at the high point of the graph, the solution was clear / yellow. And that's where there was a lot of I- floating around, and then the low points are the blue phases of the reaction when the I2 concentration was higher. So for the machine learning results, the conditions suggested by the initial procedure, which was 8.6% hydrogen peroxide and 22 degrees Celsius, ended up with a reaction length of 307 seconds, but then the reaction, or the initial conditions that the machine learning program suggested ended up resulting in a longer reaction time. So that was very exciting.
So these are the antioxidant study results. So when we saw the cinnamon tea, which was really exciting, is what we noticed is that After we collected all of our data and put it on a graph, the orange is the blank, so when there's no T suspension added, and the red is when there is a T suspension, and the arrows point to the start of the second blue phase. What we can clearly see is that there is an extended period and a longer period at the start of the second blue phase when compared to the blank. And this is because when we were talking before, when the antioxidants are donating one of their electrons, it takes a longer time for the equilibrium to be shifted in the other direction and for the next phase to actually occur. That's the same thing that we saw with the green T2. This is the start of the second blue phase where the arrow is pointed towards the green line, and then next is the blank underneath. And what we do see is a significantly smaller period with the blank than there is when the green T suspension was added. With the cranberry tea, actually, it was surprising to us. We didn't see as much of a difference as we saw within the other teas. It looks kind of similar in terms of the periods, so the amplitudes do shift a little bit, but our data at the end suggested a little bit otherwise. So this is the culmination of all of our trials and all of the blank trials, and what we can see in here to the left is that the average period for each T is really different from the average period of the blank when no T suspension was added, and this does really clearly show that there is, I guess, a significance of the T suspension being added, and it was a great way to honestly quantitatively measure how the ability of certain antioxidants and also show which T had the highest antioxidant capacity. One thing to notice is, though, that our standard error does indicate that we need to do more trials for our test to be significant. However, we are sure, since we were doing this in a classroom setting, that if we had more time and more trials were conducted, what we see observably would also show in our data.
So finally, for our conclusions and future work, for the machine learning conclusions, we did demonstrate that the machine learning program was able to successfully predict the best initial conditions for the Briggs-Rauscher reaction. We would have also liked to have more time to run more trials with that so that we could have the value stabilize even more and get more significant results. In terms of future work, in addition to running more trials, we would also like to include a multivariable regression line, because right now we just had a few single variable lines, which was great because it could evaluate each of the data points or each of the conditions by themselves, but in a multivariable one, those conditions would be linked, which would probably be more useful. And in addition, we would like to include the potassium iodate concentration in our study.
For the antioxidant studies, what we concluded was, because of the period lengths, we saw that the cinnamon tea had the highest antioxidant capacity, followed by the cranberry and ending with the green tea. And as we said before, more trials would honestly validate our findings even more. However, what we did see is that there's a lot of, I guess, potential practical implications to studying the antioxidant part of this. A lot of it has a big effect on our diet, so if you're able to see what antioxidants do well and how they can prevent certain diseases that are caused by an excess of free radicals, you know what you're putting in your body and if it's going to be helping you, and also if it's going to be preventing certain malfunctions in the future.
So basically, why does this matter? Just to summarize, as Shaezmina kind of mentioned, the ability to quantitatively measure the antioxidant capacity of these different teas is very significant, because normally previously we're able to just know if it has antioxidants or not, but through the Briggs-Rauscher reaction, because it is such a sensitive reaction and such a unique oscillating reaction, we're able to figure out exactly how great the antioxidant potential is, which will be useful later in medicine, maybe addressing some of the diseases that Shaezmina mentioned that free radicals are linked to causing. Mm-hmm. And also our interdisciplinary approach was unique in that we kind of used machine learning to figure out the best way to start our experiment, and then we're able to just kind of do a normal lab thing to do the antioxidant study.
Thank you so much for listening, and we welcome any questions.
Thank you very much. That was very, very interesting. And I have two questions. One, I'm just wanting to make sure I'm understanding the machine learning piece, that when you say it gave you your initial conditions, mm-hmm. right, the optimal conditions, mm-hmm. did that also give you the concentration dilution rates of the teas that you were testing?
So we didn't actually incorporate the teas into the machine learning portion.
Okay.
We arrived at the dilution rates after we didn't dilute the teas, and then the reaction stopped immediately after putting them in, and then we realized that we did need to dilute the teas as well.
Yeah.
Okay.
The machine learning program just gave the optimal conditions for the hydrogen peroxide solution and then the temperature.
Yeah.
Okay.
And that was when we were running the blank reactions,
Mhm.
and then we added the tea suspension after some trial and error,
Yeah.
figuring out what was the perfect time to dilute it so we could see some changes if there were any.
Okay, very good. And then my second question is, since you've done this in the laboratory, I'm curious to then know if those teas help me. Would I drink the tea and then you'd have to like check my blood to see if the antioxidants had the great effect?
That's a good question. So kind of for the future implications of this research, we're not sure exactly how like you could incorporate that, like actually seeing how it interacts in the body.
Yeah.
But there is an important part when we like antioxidants and access of those free radicals when you talk about even something like cancer or oxidative stress within your body. So a lot of the ways that I don't think you need to inject anything in you, but knowing what you're putting into your diet and which one does have a higher antioxidant capacity is, I think, important when we are talking about seeing, your free radicals and seeing how many of those you have. And it's also a chain reaction if you're not being able to donate extra electrons to them. And also I think the big part of using the Briggs-Rauscher reaction to do this is that it can quantitatively measure the antioxidant capacity, whereas before you just are told that there's antioxidants in this, so eat this. Whereas at this time, this reaction can just tell you, observably, this is the voltage and this is where you're seeing a larger period length, so this is where the antioxidants are reacting most and how they're clearly changing the reaction length in the phases.
So what do you see as a logical next extension of what you've discovered?
I think a extension for this would first of all to be trying this with more foods, because we know that teas have antioxidants, but if we're trying to develop new dietary new diets, stuff like that, taking things that we really don't know about their antioxidant capacity and using the Briggs-Rauscher reaction to do this would be helpful. And also, I think, refining our technique in this and finding a way to really use the Briggs-Rauscher reaction to, we were able to see it graphically that there was a change, obviously, but if we had had more time in the lab, we would have really liked to develop a test almost to be able to really, have a very strong measure of the antioxidant potential.
Thank you.